Good evening, ladies and gentlemen. Welcome to our 23rd award ceremony of the Eppendorf Award for Young European Investigators. My name is Andrea Ablasser, and I just briefly guide you through this evening here tonight. And it's my pleasure to officially open this award ceremony here again at the EMBL Advanced Training Centre. It's always a pleasure to be here. Thank you very much for giving us the opportunity.
Thank you very much. It's actually a great pleasure to introduce you, give you a little bit of background of the work of our prize winner from the viewpoint of someone who is not so close to the field. I'm a neuroscientist, and so I work on completely different things. Now, it is one of the main jobs of our immune system is to detect and eliminate potentially noxious external material, in particular infectious agents like viruses and bacteria. Indeed, our immune system, as we all know, is critical for survival in an environment where we are essentially constantly under attack by pathogens. How the immune system manages this complex task never fails to fascinate me. And as I said, I'm a neuroscientist, and I've always thought the brain is the most complex organ in our body, but I'm starting to have second thoughts. If you see, the amazing complexity of the immune system and the various pathways unraveling, and there are still many, many surprises coming up. Almost every month if you follow the journals. Now, mammals possess two kinds of immune system responses termed innate and adaptive immune response. The adaptive immune system is tailored to cope with each hostile organism in a very specific manner, and it has almost extraordinary features and has been in the center of the last decade to really understand better how the adaptive immune system works. And it's not only defending providing a very selective defense against invasive organisms, but it can also be utilized to fight diseases. For instance, we all, I guess, know about the kind of the buzzword immunotherapy of cancer. It has really kind of major inroads into previously incurable diseases. However, this immune response requires time. It takes days to weeks until we really can mount a full-blown adaptive immune response. And if this were our only protection from pathogenic agents, we would be quickly overwhelmed by fast replicating organisms. So this is not enough. So the innate immune system has a much shorter response time and is our first line of defense in this case. Now, in contrast to the high specificity of the adaptive immune system, the innate immune system or the response relies on a kind of standard arsenal of defense weapons that are ready at any time. So they are the innate immune system targets features which are common to many pathogens instead of being very specific. So the only condition is essentially that these features are sufficiently distinct from our own body's signals to be, let's say, recognized and then to mount an immune response. Targets recognized are, for example, surface proteoglycans shared by many bacteria. Surprisingly, it has become apparent in the last decade or so that various nucleic acids, including double-stranded DNA, can actually trigger a response of the innate immune system. Now, at first glance, this doesn't make sense. All our body cells contain DNA, and DNA is, in a way, you cannot hide DNA from by making tricks with proteins where you can just change surface glycosylation, as many viruses have done. So how can this be a functional signal for the immune response? Well, the answer is relatively straightforward. DNA, double-stranded DNA, is normally in a healthy cell is supposed to be packaged in the nucleus. So if there is DNA in the cytoplasm, it's usually, the chances are very high that it's something you want to combat, that is coming from a pathogen. And this is essentially what this response is based on. Now the question is, how do you recognize this DNA in the cytoplasm? Now, how do you trigger the response? Now, it is known that there are quite a few DNA-binding proteins in the cytoplasm. It was known for some years. And it's also known that the response which is triggered is essentially an inflammatory response. So the cell which is affected secretes cytokines and other, let's say, signals into the environment, signaling that there is a problem here and that you get an inflammatory reaction. But the question is, how can DNA-binding proteins tell the cell to synthesize and secrete cytokines? And this mechanism has only been uncovered very recently, and this is where the work of our prize winner has been indeed instrumental for clarifying these steps. Now, Before Andrea tells you more about her work, let me just briefly introduce her background. Andrea grew up in a family of medical doctors and became fascinated by science already during the early high school years, from what I've heard from her. And then in a family of doctors, medicine was probably the obvious choice, and she took up medical studies at the University of Munich. But she told me that she was unsure originally where to go, and also took up physics courses, and the interest in science was apparently one of the features that distinguished her from many other medical students who only want to know how to treat patients. And while pursuing a medical degree, she became more interested in the scientific foundations, and everyone who knows medical education, and those of us who teach, I did for some years, know that the problem is you cannot really go in too deep into the scientific foundation of medicine. And for these reasons, she decided early on to, after her intermediate exams, to join a scientific lab, and she ended up in the Department of Clinical Pharmacology headed by Stefan Endlers at the University of Munich. And there she met a postdoctoral scientist, Veit Hornung, whom she followed for a brief stint to the University of Massachusetts before she then learned his team when she became a professor at Bonn University, I think as the first postdoc in his team, and then worked with him for some years. In the lab, they were apparently hot on the trail of this kind of DNA recognition signaling and trying to identify the missing link between DNA sensing and cytokine activation. And what I've heard from you is that you had a bit of bad luck, apparently, at the very beginning. They were scooped, because apparently another lab, the lab of Jason Chen, found out that there is a very unusual dinucleotide serving as a signal, which has a name, it's kind of a dinucleotide formed from GMP and AMP called CGAMP, as a key intermediate. They also showed that the molecule is synthesized by novel and hitherto uncharacterized DNA binding protein, now termed CGAMP synthasase, or CGAS, is a biosynthetic enzymes. Now, when you're scooped, you can say, well, shit, yeah, problem solved. Well, this is not what they did. Instead of giving up, the team set out to work on the new pathway, and then the surprises kept on coming. Andrea's first major discovery was that the structure of CGAMP, which was known from bacteria, was actually not identical. And that was actually critical to understand its biological activity, because only in this kind of mammalian with a different chemical structure, only this kind of version was really active in triggering the immune response and in binding and activating its its target protein, which is a homodimeric membrane protein with a weird name over the complicated name, ER-resident protein stimulator of IFN genes, abbreviated STING. Then Andrea found out that the novel second messenger does not only activate intercellular mechanisms, but also spreads to other cells. It serves kind as a fire siren to really alert this environment on this infection, or on whatever this kind of DNA is coming from. These and other discoveries resulted in an extremely impressive list of high-ranking publications from a postdoc that I've really only rarely seen. This is very impressive. She also has received some awards, like the Max von Pettenkofer Prize, the Jürgen Wieland Prize, the Paul Ehrlich and Ludwig Darmstädter Prize for Young Researchers, and the GlaxoSmithKline Award for Basic Medical Research. In 2014, Andrea became a tenure-track assistant professor at the EPFN Lausanne. You heard this already. Since then, her performance as a group leader has been at least as remarkable as before. Despite the short period of time, she has already made seminal contributions. She showed that the pathway is also activated in senescent cells by its own DNA, because the DNA starts to get into the cytoplasm when the nuclei become destabilized. And she has also identified the first inhibitor of the central mediator STING with remarkable clinical potential, which is now in press in Nature. The first paper was published in Nature Cell Biology, which I'm sure will please our partners in this business. Now, I don't want to say anything more. Let's welcome our this year's prize winner, Andrea Ablasser.
Okay, so hello everyone. First of all, thank you very much for being awarded with this prestigious award. It's really a great honor to receive it, and I'm feeling very flattered to receive this prize. So we're working on intracellular DNA sensing mechanisms, and actually what you're seeing here, this is a cell, and in red you see STING, which we just heard is a central component of this sensing pathway. And what I'd like to do now in the next 20 minutes or so to give you a little bit of an introduction and explaining what actually happens in this cell when it sees double-stranded DNA. So just to get started, I think we've heard it. One of the most fundamental questions in immunology is, how do we sense infection? How does our body sense that pathogens are present? And it turns out that one important mechanism to do so is that the cells, the innate immune cells, they have evolved certain receptors called pattern recognition receptors that are present on those cells, either the cell membrane or intracellular, and they're specialized to detect pathogen-associated molecular patterns called PAMs, and these PAMs are very specific for microbes, so they're usually not present in host cells, and this allows the immune system to straightforward distinguish self from non-self. There's a little bit of a problem here, and I'll talk about this later. But in general, so this is what we call pattern recognition sensing. And that's really critical because these receptors, they're hardwired to immune effector responses. And what these cells then do, they are able to directly, through the activation of pattern recognition receptors, kill microbes. So innate immune cells are able to, for instance, in lysosomes, degrade pathogens. But they're also essential to instruct the second branch of the immune system, the adaptive immune system, which then more specifically kills microbes through, for instance, the production of antibodies. Now, what has been known for quite some time, discovered in the early 60s, that foreign nucleic acids, viral-derived nucleic acids, RNA and DNA, are very, very potent immune stimulatory molecules. And these are actually the entities that our cells sense when they sense viral infection. And this is largely to the fact that viruses, they often lack very specific patterns. They replicate in host cells, so they lack often specific molecule structures that allow the immune system to distinguish straight away self from non-self. So these are the entities that are usually recognized. And how does this work? Well, we've just heard about the major players. The predominant sensor of double-stranded DNA inside cells is called cGAS. It's an enzyme. It binds to double-stranded DNA and then produces the cyclic dinucleotide called cGAMP, which we described chemically. So it's an asymmetrical cyclic dinucleotide. It's a very potent activator of STING, which we've just also heard. It's a critical molecule localized in the endoplasmatic reticulum, and upon binding cGAMP, STING then recruits kinases and essentially drives inflammation through de novo transcription. Now, what's interesting about the signaling cascade, if you think about it, it's a cascade that has two receptors, A and B, which are connected through a soluble cyclic dinucleotide. And we've discovered that this allows for quite unique signaling mechanisms, because what we've observed is that cGAMP itself can be transferred to bystander cells and alert bystander cells of ongoing infection. And what you're looking here is actually cells. In blue, you see the nucleus. In red, you see STING. And this is just a control. So STING is in the ER. Once STING is activated, it forms these dots or bubbles or specks, and you can record STING activation in live, in living cells. And what we did, very naive initially, was just to overexpress cGAS, and then we thought, well, we can activate STING in these cells. But what you see here, quite impressively, is that there is STING activation also ongoing in the cells that are in immediate contact of the initial cells. So indicating that there is some information being transferred from this cell to this cells. And you can actually also see this in life. So in what you see here, this is a cell labeled with a dye, and this dye can be transferred to other cells, and this indicates that they're connected. And what you're seeing here is that once the cell spreads a dye, you see that here STING is activated, and I should tell that in this cell, cGAS is activated. So what you can see here is how really cGAMP spreads and activates STINGs in bystander cells. So, and what we concluded here is that cGAS is an enzyme that also allows for horizontal transfer of signaling molecules and alerting bystander cells. Now, it turns out that despite its very recent discovery, so at the beginning of 2013, it turns out that cGAS is a major player, a major receptor for many distinct medically relevant pathogens. So, this list is ever growing. A lot of DNA viruses are dependent on being sensed, or our host, our organism is dependent on cGAS in recognizing many DNA viruses, also RNA viruses, dengue virus, West Nile virus. Dendritic cells, macrophages, activate cGAS in response to retroviruses, including HIV. With my laboratory, we've shown that cGAS is also activated in the context of Mycobacterium tuberculosis infection, which is the causative agent of tuberculosis, and many other pathogens. So it's a key sensor for many distinct pathogens. And it's a very potent immune stimulatory molecule, cGAM, and that intrigued now many companies actually to use these cyclic dinucleotides to boost anti-tumor immunity, and we've just heard it. So checkpoint blockers are really transforming medicine and transforming cancer therapy, but it's just that only in a fraction of patients are these checkpoint blockers active. And probably what they lack is they're working on the adaptive immune system. And the question here, the big question is now, by adding a component of the innate immune system, for instance, cGAMP, can we then boost this anti-tumor immunity and maybe even enhance the potency of checkpoint blockers. And actually, cGAMP derivatives are already in clinical trial to exactly test this hypothesis. Now, this is what is a very key feature about cGAS. So it binds to double-stranded DNA sequence independently. And actually, what I've just told you in the introduction is that pattern recognition receptors distinguish self from non-self. This really fundamental rule is actually violated in the case of cGAS because it binds to double-stranded DNA irrespective of sequence and origin. And what this affords is that our immune system has other accessory molecules, accessory mechanisms that allow them to distinguish self from non-self or dangerous from non-dangerous. And one key accessory mechanism is the use of nucleases. So nucleases, DNases, are constantly active in cells to dispose of self-DNA. And once these nucleases are mutated, cGAS becomes activated and triggers inflammation. And this is critical because there are mutations in these nucleases, which I've just shown you, that cause detrimental disease. So there's a very rare disorder called Aicardi-Goutieres syndrome that mimics that, and on a brain scan looks as if these children were infected in utero with a virus. But what they actually have is they have aberrant activation of cGAS due to their malfunction of these DNases. And it's a very severe disease and currently cannot be cured. This is a mouse model of this Aicardi-Goutieres syndrome where they have a mutation in this nuclease, and these mice die shortly after birth because of a very dramatic inflammation in the heart. So this is just to illustrate what cGAS can do when it sells self-DNA. Now, nuclease is one. Activity of nuclease is one solution. The other solution is compartmentalization, so constraining DNA in locations where cGAS is not present. And I just will show you a recent finding of our lab where we've shown that this loss of compartmentalization can activate this pathway in the context of cellular senescence. So what is cellular senescence? So cellular senescence is a stress response It's a program that's activated when cells are constantly exposed to distinct stressors. And these cells, once they enter into senescence, their permanently growth arrested, meaning that they can no longer proliferate. But they're still producing cytokines. And this production of cytokines has major biological functions. It contributes to tumor suppression. Senescent cells are involved in embryonic development and wound healing. But these senescent cells at the same time are detrimental in aging and contribute to age-related diseases. So what's interesting is that during cellular senescence, the nuclei shows certain alterations. And these alterations, what you can see here, this is a senescent cell. In red, you see lamin B1, which is a filament beneath the nuclear membrane. And what you see in gray is chromatin, so DNA. And what you can see here is that there is chromatin leaking or blebbing out of the nucleus. So, and this was very intriguing. And what we've gone on to show is that actually CGAS senses this chromatin herniation and then is activated and actually contributes to the production of this inflammatory phenotype of senescent cells. So, but what we're, and just very briefly as a follow-up, what we're now intrigued is to understand how this DNA leaks into the cytosol. At the first, defective nuclei and then activate CGAN. But if you look very closely, this is actually quite complicated. So the nuclear envelope is actually tethered and connected to an actin filament. So it's mechanically coupled to the cytoskeleton. And DNA is in here, lamin B1 is in here. And the real question that we're asking now, how does this blebbing occur? And we've studied this very systematically. And what I just want to show you is that, in fact, what CGAS senses is aberrant nuclear mechanics. So the nuclear envelope and its coupling to the cytoskeleton is critically to trigger this response. And what you're seeing here is a movie, and in green you see a nucleus. So this is a protein that is just able to reside in the nucleus. So you don't see the cytoplasm of the cell; it's dark. And in red you will see CGAS. And when I started this movie, what you see, so this is the nucleus, it's intact, and what you suddenly see in a minute is that there was a loss in signal, meaning that there's a rupture. And along with this rupture, CGAS entered into the nucleus and binds to DNA and becomes activated. So what we think is actually CGAS, it's a sensor of excessive biomechanical stress to the nucleus and transforms this into an inflammatory response. Now, why is this important? Well, we know that there are several disorders, genetic disorders, that are linked to accelerated aging syndromes, progeria syndromes, that are linked to having mutations in essential genes that form the nuclear envelope. And we think that there's a link between CGAS. So we think that actually CGAS might play a role in the pathogenesis of some of these very rare diseases, and probably also during aging. Now, this brings me to the last part of my talk. Because of its detrimental role in these diseases that I just mentioned, it's intriguing to think of targeting this pathway specifically pharmacologically to intervene into the pathogenesis of these diseases. And we get intrigued by this idea. And there are many diseases, actually, and we begin to understand that self-DNA sensing is pathogenic in many distinct disorders. So I've mentioned Aicardi-Goutieres syndrome, a very rare genetic disease. There's another clinical syndrome in which STING has mutations that render it hyperactive. Both these diseases are kind of very severe, and there's so far no real cure. What we also begin to appreciate is that the CGAS-STING pathway is relevant in a much broader context of autoimmune or classical autoimmunity, such as systemic lupus erythematosus. We know there was a study recently that showed that probably 15% of patients with SLE have elevated cGAMP levels, so which is indicative of of at least some activation of this pathway. But what's even more intriguing that there are many more complex disorders where in mouse models, cGAS has shown to be detrimental. Myocardial infarction, age-related macular degeneration, sepsis. And I just wanted to show you one piece of work from a colleague. So this is a study performed in mice. They had a myocardial infarction model, and in this model, about 50% of the mice die because of myocardial rupture events. And this is a mouse that lacks cGAS, and you can see in this group, nearly all mice survived. So indicating that blocking this pathway might, be beneficial in the context of myocardial infarction. And how does this work? Well, the mechanism that was proposed is that in myocardial infarction, there's a synchronized cell death. So at a certain unpredictable time point, many cells suddenly undergo cell death, which leads to an accumulation of self-DNA, and this overwhelms our normal system. And this huge accumulation of self-DNA then triggers the cGAS-STING pathway, drives inflammation, and contributes to an inflammatory response in the myocard, which is then detrimental. Now, because of this, we get interested in targeting this pathway. And we've actually started all this work before STING was, or at a time where STING was, or the pathogenic function of STING was not at all known. So we just actually wanted to get tools to study this pathway in the lab. So we set up with a chemical screen. We screened thousands of compounds in a cellular assay, and what we found is quite intriguingly two very potent compounds. And what you see here is this is a dose response, and this is STING, so it's nicely inhibited, and here controls are not inhibited. So we were kind of very, we thought we had very good compounds. Again, this is a cellular assay, so we're looking at a very downstream response. And we confirmed this in many distinct primary cells, so mouse, macrophages. It worked beautifully. But then we went into human cells, and unfortunately, these compounds did not at all block human STING. And maybe, you can appreciate that at the beginning we were kind of a little bit frustrated, but in the end, this was actually a fantastic result, because what it told us that these compounds would really target STING. I mentioned to you before, we screened cells, we had a cellular readout, and as you may appreciate, those compounds could have many distinct effects. But what it told that it works on mouse STING, but not in human STING, told us that these compounds really act on STING itself. And this actually greatly facilitated us to understand how these compounds work. So this is a model of STING. We've just heard it's an ER membrane protein. It's a homodimer. This is the ligand binding domain where it interacts with the cyclic dinucleotide. This is the transmembrane domain. And what we've done is we mutated every single amino acid in this transmembrane domain, because we've, by the species specificity, we found out that this molecule actually works through this part. And it's this is not crystallized, this part, it's completely unknown. But by doing so, what we found is we found one cysteine residue, which once mutated, is no longer sensitive to this inhibitor. And by many follow-ups, what we've shown is that actually the cysteine undergoes a covalent interaction with this molecule, and thereby STING is blocked. Now, we had compounds with their work in vivo. So this is a mouse model that we used. It's a TREX1 knockout. Maybe you remember this was the mouse that had this severe inflammation in the myocard. So we treated this mouse 3 months with this compound, and then we looked at the heart and to assess the inflammation. And it was quite intriguing. So this is the heart of the mouse that was treated with the control, and as you can see, the blue dots, this indicates cell, and this is heavily inflamed myocard. And when you treat these mice with the inhibitor for 3 months, the heart looks essentially quite healthy. And this is the scores, untreated, treated, untreated, treated. Systemically in the heart, smooth muscle, stomach, and tongue, we can suppress this inflammation. So we were quite excited about this result, but still, we just had mouse compounds. So we then derivatized these compounds and did further screens, and what we indeed found, we find now a very potent human compound. These are human cells in which we can block STING. And that's kind of the summary, and then as we just heard, this paper is now in press. So we've, for the first time, shown the first-in-class small molecule inhibitors of STING, and proved it as a proof of concept that we, by pharmacologically intervening into this pathway, can ameliorate autoinflammatory disease in mice. Okay, so with this, I come to the most important slide. Of course, it's I've been throughout my career very fortunate and privileged to work with excellent people. So this is my group, and I've a fantastic team that's very passionate, very exciting about research. And the two stories that I told you, so in the senescence work, this is an extremely talented student, PhD student, that after 1 year in my lab had this discovery and worked it through. It's quite impressive. So that's Céline Gluck. And the inhibitor paper was also worked out by another PhD student, Simone. And then another great team of postdocs and further students. I'd also like to highlight our collaborators, our chemists. We worked together with colleagues in Tübingen and Oxford for a senescence model. And of course, my supervisors, Veit, Hornung in Munich, Gunther in Bonn, and also Stefan Andres in Munich. They really intrigued me, and I'm very grateful for them. For their mentoring and supervision along the way. Thank you very much.